Hey everyone, welcome back to ASCP Preparation Camp. Today we're diving deep into something that might seem simple at first glance, but trust me, it's one of the most powerful and adaptable systems on the planet. We're talking about bacterial genetics. Now, bacteria don't just survive, they evolve at lightning speed. And in this episode, we're going to explore the precise architecture of their genetic code that makes all of this possible. We'll uncover how these microscopic powerhouses manage their genetic material, why they're so unbelievably good at resisting the strongest antibiotics we throw at them, and the surprisingly elegant switches they use to regulate their entire survival strategy. The information I'm sharing today is pulled from deep, detailed sources in microbial genetics, and we're focusing on the core blueprint for rapid bacterial evolution, things like mobile DNA elements, unique mechanisms of gene transfer, and the processes that aren't just academic curiosities. These are the foundations of global infectious disease threats that you'll definitely encounter on the ASCP exam. So let's get started. If there's one critical takeaway from today's episode, it's about speed. The sheer ability of bacteria to transfer critical genes, especially those carrying resistance blueprints, not just to their offspring vertically, but horizontally, sideways, across species in real time. This is the reason antibiotic resistance is such a dire public health emergency right now. But before we understand how bacteria share genetic information, we first need to understand what they're sharing. And to appreciate that speed, we have to look at their infrastructure. The bacterial genome is a masterpiece of compact design, built for hyper-efficiency. Bacterial genome architecture. Let's unpack this compact architecture. The bacterial genome breaks down into two main components. First, the chromosome. Think of this as the main operating system. It's a single, very long circular piece of double-stranded DNA carrying all the essential genes. We're talking about 2,000 to 4,000 genes, roughly 3,000 kilobases of DNA, all tightly folded and packed inside the cell. If a bacterium loses its chromosome, it dies. Instantly. Non-negotiable. That's the core hardware. Second, plasmids. These are the optional accessories that give bacteria a competitive edge. Plasmids are small, independent circles of double-stranded DNA, much smaller than the chromosome, less than a tenth the size, maybe 5 to 100 kilobases. But they are the true secret weapons. Crucially, they replicate totally independently of the chromosome. A single cell can carry anywhere from 1 to maybe 20 copies of a single plasmid. And while plasmids carry what we call non-essential genes, those genes are often the difference between life and death in a clinical setting. They carry the blueprints for antibiotic resistance and the ability to cause disease. Think of them as the ultimate optional survival apps. Now, here's where the architecture gets really fascinating for anyone tracking infections. Bacteria often incorporate highly specialized additions called Pathogenicity Islands, or PIs. These are massive, discrete genetic segments, 10 to 200 kilobases long, that encode a pathogen's entire deadly toolkit. We're talking about potent toxins, adhesions for sticking to your tissues, complex secretion systems to inject virulence factors into your cells, and even iron transport proteins to steal your nutrients. They can take a relatively harmless bug and turn it into a high-grade killer. But the critical insight here isn't just what they do, it's where they came from. PIs are almost always acquired through horizontal gene transfer, or HGT. That means bacteria stole that technology from another organism, maybe even a different species, rather than inheriting it from an ancestor through normal vertical transfer. So how do we know they were stolen? There's a dead giveaway. If you analyze the DNA sequence of a pathogenicity island, it will often have a dramatically different ratio of guanine and cytosine bases, the G plus C content. It's like finding a paragraph written in ancient Greek suddenly embedded in a textbook written entirely in English. You know immediately it was smuggled in. PIs are smuggled technology, and sometimes the smuggler itself is a virus. That brings us to our next key player, the viral engineers we call bacteriophages. A bacteriophage, or just a phage, is simply a virus built to infect and replicate inside a bacterial cell. Structurally, they're minimalists, just nucleic acid, DNA, or RNA, tucked inside a protein coat. Many phages operate in extreme violence mode, the lytic cycle, or virulent takeover. Let me walk you through how fast this is. The clock starts at attachment, T equals 0. The phage latches on, then immediately injects its nucleic acid into the cell, leaving the protein shell outside, totally empty. Unlike viruses that infect our cells, phages don't bother bringing the whole code in. That saves precious seconds. Within just 10 to 25 minutes, that phage has completely hijacked the cell's machinery and is synthesizing hundreds of new viral particles. By 30 minutes, a phage enzyme dissolves the bacterial cell wall, leading to catastrophic lysis. The cell just explodes, releasing hundreds of new phages to start the cycle all over again. It's a literal blitzkrieg, and you can actually see this devastation in the lab. If you put a single phage on a lawn of bacteria, you'll see a plaque, a visible clear spot where that one particle has destroyed millions of cells in just a few hours. But not all phages are immediately destructive. Some are temperate phages, and they have a choice. Instead of immediate lysis, their DNA can integrate directly into the host chromosome, becoming a genetic sleeper agent. This integrated, dormant DNA is called a prophage, and the cell is now called lysogenic. In this state, a regulatory protein represses all the phage genes. The host survives, functions normally, and just copies the phage DNA every time it divides, for potentially thousands of generations. Incredibly stable. But here's the catch. It's a time bomb. The trigger is usually DNA damage or severe stress, think UV light or certain chemicals. This stress destabilizes the repression. The prophage cuts itself out of the chromosome, starts replicating on its own, and kicks off the lytic cycle. This process is called induction. The phage basically waits until the bacterial neighborhood is collapsing, then makes a break for it. Smart, right? But even better for the bacteria, while that prophage is dormant, it can grant new powers. We call this lysogenic conversion. The genes carried by the integrated prophage can make the bacteria more virulent. A classic example is the toxin produced by the bacteria that causes diphtheria. Without the prophage, the bacterium is harmless. With the prophage, it becomes deadly. It's the ultimate biological hitchhiker. Which brings us right to our next major topic. How do these powerful genes, resistance factors, pathogenicity islands, dormant prophages, actually move between cells? This rapid exchange, horizontal gene transfer, is the core of bacterial adaptability. And there's a basic rule here: any transferred DNA that doesn't have its own origin of replication has to integrate into the recipient's chromosome to survive. If it can't, it just gets diluted out, with the exception of plasmids, which carry their own origin of replication. Let's run through the 3 ways bacteria swap genetic blueprints. Method 1, conjugation. The most direct method. This requires direct cell-to-cell contact. You have a donor cell, the F+, and a recipient, the F-. The donor has special equipment called the sex pilus. This hair-like projection reaches out, attaches to the recipient, and forms a physical cytoplasmic bridge. It's like docking a cable between 2 cells, a biological handshake that turns into a dedicated data line. What passes through? Typically a plasmid. One strand of the double-stranded plasmid is nicked or cut, and as it peels off, it passes through the bridge to the recipient. But here's the elegant part: simultaneously, both cells synthesize the complementary strand. So in the end, you have 2 complete double-stranded plasmids, one in the donor and one in the new recipient. It's a highly sophisticated one-way copy and paste operation that ensures the resistance gene, or whatever it carries, is now doubled in the population. Method 2, transduction. This one uses the phage as a delivery vector. Transduction is gene transfer by a phage. No cell contact needed. The huge benefit is that the DNA is protected inside the phage's protein coat. It's safe from any DNA-degrading enzymes out in the environment. We have 2 flavors of this. First, generalized transduction, which is completely random. During the lytic cycle, the phage is chopping up the host's chromosome. By sheer accident, a random piece of bacterial DNA gets mistakenly packaged inside a new phage coat instead of the viral DNA, a simple packaging error. If that faulty particle then infects a new cell, it injects the bacterial DNA instead of viral DNA, and the recipient can be transduced. The second flavor, specialized transduction, is much more targeted. It only transfers specific bacterial genes, the ones located right next to where a prophage was integrated. It happens when the prophage cuts itself out of the chromosome and accidentally snags a little bit of the adjacent bacterial DNA along with it. Method 3, transformation, the uptake of naked DNA. This is passive uptake, but it requires a special physiological state called competence. When a bacterium dies, its DNA can break up and just float around in the environment. A competent cell can sense and grab this free-floating DNA. It's genetic scavenging. As the double-stranded DNA enters the cell, one strand is usually destroyed. The remaining single strand can then invade the resident chromosome and swap out existing genes for the new ones it just found. This process, discovered back in 1928, is what confirmed DNA as the genetic material in the first place. Clinically though, it means bacteria are constantly picking up new instructions from microbial debris. This constant transfer, conjugation, transduction, transformation, ensures bacterial populations are never static. And this variability is fueled even more by internal engines of change, random mutations and, critically, mobile genetic elements. Let's focus on the heavy lifters of variation, transposons, the jumping genes. Transposons are true genetic anarchists. They're segments of DNA that have the ability to excise themselves and just jump to new locations, on the chromosome, between plasmids, wherever. They often carry genes for the enzyme that lets them jump, called transposase, and very importantly, an antibiotic resistance gene. They're almost like genetic viruses themselves, just without the protein coat. Their movement is governed by short inverted repeats at their ends. We classify them as either replicative, which leaves a copy of itself behind so it spreads, or non-replicative, which just moves from one spot to another. The clinical link is inescapable. Transposons are why resistance spreads so fast. A transposon with a resistance gene can jump onto a plasmid, that plasmid gets conjugated into a new bacterium, and then the transposon can jump off the plasmid and onto the new host's chromosome, making it a permanent fixture. It's the ultimate adaptability engine. And this forces us to detail the 5 primary mechanisms bacteria use to build their arsenal against our drugs. Let me run through them quickly. First, decreased permeability or uptake. They alter the gates to the cell. In gram-negative bacteria, for example, they can change the size of the porins in their outer membrane, basically shutting the door on drugs like beta-lactams or tetracycline. Can't get in, can't do damage. Second, antibiotic efflux, an active removal system. Think of these as super-efficient bilge pumps. They're multi-component membrane systems that recognize toxic substances, including antibiotics, and pump them right back out before they can even reach their target. Third, alteration of the target site, changing the lock so the key doesn't work anymore. A famous example is Streptococcus pneumoniae, which alters its penicillin-binding proteins, or PBPs. The antibiotic can't bind effectively, and it loses its power. Fourth, inactivation or destruction of the antibiotic, a direct counterattack involving enzymes that chemically destroy the drug. The most notorious are the beta-lactamases, which shred the crucial beta-lactam ring in penicillins and cephalosporins, rendering them completely useless. Fifth, acquisition of a new target, not just changing the lock, but building a whole new door. Take Staphylococcus aureus resistance to vancomycin. Vancomycin normally targets a structure in the cell wall called diala-diala. Resistant staph acquires genes that change that structure to diala-dilac. Vancomycin can't bind the new target at all. Drug is useless, cell wall is perfectly healthy. It's pure evolutionary chess. But all this resourcefulness, making toxins, building pumps, must require a massive amount of energy. Bacteria can't afford to be wasteful, which brings us to their sophisticated switchboard, gene regulation. Most of this happens by controlling transcription. The classic blueprint for this is the lac operon, which demonstrates negative control. The problem to solve is simple. You only want the genes to break down lactose when lactose is actually around. It's a cost-benefit analysis. When lactose is absent, the cell doesn't want to waste resources. A repressor protein binds to a spot on the DNA called the operator site and physically blocks RNA polymerase. The operon is off. But when lactose is present, lactose itself acts as the inducer. It binds to that repressor protein and changes its shape, inactivating the repressor. It lets go of the DNA, the road is clear, and transcription begins. The operon is on. Simple on, off switch. But it's actually a double-check system. Bacteria always prefer glucose over any other sugar, and this preference is managed by a second layer, positive control, or catabolite repression, the prioritization system. To really use lactose efficiently, the lac operon needs an accelerator pedal. This is an activator protein called CAP. CAP has to be bound near the promoter to really recruit RNA polymerase effectively. But CAP is only functional when it's bound to high levels of a molecule called CMP. And the cell chemistry is set up so that high glucose means low CMP. So high glucose, low CMP, CAP is inactive. The operon stays mostly off, even if lactose is there. The cell is saying, why bother with the complex sugar when the easy one is right here? But if glucose is low, CMP spikes, CAP is activated, and if lactose is there, the operon roars to life. It's a beautifully layered switch. And that system of individual regulation scales up globally. In response to extreme stress, like starvation or heat shock. Bacteria can perform system-wide resets by swapping out the normal sigma subunit of their RNA polymerase for an alternative one. This new sigma subunit recognizes a completely different set of promoters, causing a coordinated change in thousands of genes at once. Incredible. So as we've seen today, the bacterial genome isn't static at all. It's constantly being reorganized by jumping genes, viruses, environmental pressures, all managed by sophisticated, multi-layered regulatory mechanisms. The central key takeaway remains the sheer speed and variety of horizontal gene transfer, conjugation, transduction, transformation, coupled with mobile elements like transposons. This genetic fluidity is what makes bacteria the ultimate biological adapters, and why they're so good at surviving everything we throw at them. But here's a provocative thought to leave you with: transposons are critical for resistance, sure, but they were discovered because they inserted into and destroyed a functional gene. If these jumping genes are constantly reorganizing the genetic deck, we have to consider the non-resistance functions we might be accidentally altering. Just by changing bacterial environments, say, with novel non-antibiotic treatments or even environmental pollutants, if a transposon jumps into a key metabolic or virulence pathway, we could stimulate unforeseen evolutionary changes that go way beyond just drug resistance. We could potentially create entirely new, unpredictable pathogens. This constant genetic reshuffling, that's the real frontier we need to monitor. Alright everyone, that wraps up episode 3 of our microbiology module. I hope this deep dive into bacterial genetics has given you a solid foundation for understanding not just how bacteria survive, but how they thrive in the face of our best antimicrobial weapons. This is high-yield material for the ASCP exam, so make sure you review these concepts. In our next episode, we're going to tackle Staphylococci, one of the most clinically significant bacterial genera you'll encounter. We'll break down their characteristics, pathogenicity, and of course, their resistance mechanisms. You won't want to miss it. Now, before you go, if you're finding this series helpful and you want even more high-yield, exam-focused content to complement your study routine, I've got 2 books available on Amazon that are perfect for your ASCP prep. The first is ASCP Short Notes in Hematology, a concise, focused guide that covers all the essential hematology topics you need to master. The second is ASCP Short Notes in Chemistry, same format, same efficiency, covering all the key chemistry concepts for the exam. Both books are designed to give you the critical information you need without the fluff, so you can study smarter, not harder. You can find them both on Amazon by searching for my name, Mohamed Hamamma, or just check the links in the description below. Thanks so much for watching, everyone. Don't forget to subscribe to ASCP Preparation Camp, hit that notification bell so you never miss an episode, and if you found this video helpful, give it a thumbs up and share it with your classmates. Keep studying, stay focused, and I'll see you in the next episode. This is Mohamed Hamamma, until next time.